One small cookie, one big adventure to divide.
Let's see how it's done. The cell cycle is a series of events that take place in a cell leading to its division and replication. For the most part, the cell is an interphase. Only for the last few parts of the cycle does the cell take part in mitosis and lastly cytokinesis.
Many cells within our body go through mitosis every day. Mitosis allows for the growth, replacement, repair, and regeneration of cells. The first stage of mitosis is interphase. This is where the chromosomes duplicate and the nucleus is a dense mass of DNA. Next comes prophase. Here, the dense cluster of chromatin begins to shorten and condense to form chromosomes. The nuclear membrane and nucleolus surrounding the DNA disappears. The centrioles migrate to opposite ends of the cell. After prophase comes metaphase. Here, the chromosomes migrate to the center of the cell and align themselves along the metaphase plate. The spindle fibers begin to appear and attach to the centromeres. Next comes anaphase. Here, the centromeres separate and the previously attached spindle fibers pull the chromosomes to opposite ends of the cell. This results in twice as many chromosomes. Lastly, we have telophase. Early in telophase, the chromosomes reach the poles and begin to form nuclei. The two nuclei formed are identical to each other as well as to the parent cell. The nuclear membrane reforms, the chromosomes begin to unwind, and the spindle fibers disappear. In late telophase, cytokinesis, the division of the cytoplasm, occurs. A furrow forms and the cell is pinched into two cells, each with 46 chromosomes. In our bodies, we also have meiosis. This is a type of cell division that only occurs in eukaryotes. In meiosis, the parent cell divides and produces 4 genetically different gametes. The process of meiosis is divided into 2 phases, meiosis 1 and meiosis 2. Meiosis 1 includes the separation of homologous chromosomes. Interphase. In this phase, like mitosis, chromosomes replicate but are still a dense mass of DNA with no shape. Next comes prophase 1. Here, the chromosomes condense and migrate towards the nuclear envelope. Spindle fibers form and synapsis of the pairing of homologous chromosomes begins to take place. The homologous chromosomes interchange equivalent sections of chromatids, known as crossing over. The nuclear envelope and nucleoli dissolves. Then comes metaphase 1. Pairing of homologous chromosomes takes place in the equatorial plate, which lies in the center of the cell. Next comes anaphase 1. The chromosomes migrate to the opposite poles of the cell. Unlike mitosis, the sister chromatids are not separated but remain together. After anaphase comes telophase 1. As the chromosomes continue to migrate towards the poles, condensation of the chromosomes as well as cytokinesis takes place. 2 daughter cells with haploid chromosome number are formed.
Here we enter right into meiosis 2, which includes the separation of sister chromatids. Interphases are passed and the cell moves right into prophase 2. Here the nuclei and nuclear membrane are separated and spindle fibers appear. The chromosomes begin to move towards the equatorial plate while the 2 sister chromatids are still held by the centromere. Next comes metaphase 2. In this phase, the chromosomes are aligned on the equatorial plate and the spindle fibers attached to either side of the centromeres. Then comes anaphase 2. Here the spindle fibers pull the sister chromatids towards opposite poles of the cell. Lastly comes telophase 2. At this final stage, 4 nuclei, 2 in each daughter cell, are formed. Cytokinesis occurs and each of the 4 nuclei develops nuclear envelopes. 4 daughter cells or gametes are formed, each with 23 chromosomes.
Although mitosis and meiosis have many similar phases, they have many differences as well. For example, mitosis has 1 division resulting in 2 cells, whereas meiosis has 2 divisions resulting in 4. During prophase, there is no crossing over in mitosis, but there is in meiosis. Also, during metaphase in mitosis, individual chromosomes are arranged on the metaphase plate, whereas in meiosis, it is pairs of chromosomes that are arranged. During anaphase, the chromatids in mitosis separate, but in meiosis, they stay together. Furthermore, in mitosis, the daughter cells have the same number of chromosomes, whereas in meiosis, the daughter cells have half the number. Also, mitosis produces genetically identical daughter cells, but meiosis produces genetically different daughter cells. Lastly, mitosis occurs in all parts of the body, but meiosis only occurs in the gonads. And that is what Nicole
And Lauren
want to tell you about meiosis and mitosis.